Hi everybody. Today we're going to talk about the organization of eukaryotic genome. As the term implies, eukaryotic genome has to be organized in a very different manner unlike the prokaryotes because the size of the eukaryotic genome is being found to be very big. If you go to consider the human genome as such like that, you can see that it is made up of 3 million base pairs and they are being arranged into 46 chromosomes. And a similar eukaryote, another eukaryote called the Paris japonica, it's a plant which was being found in Paris. Why is this plant noted? It is the largest eukaryotic genome reported and it has about 150 million base pairs. And you can see that these 150 million base pairs of Paris japonica genome is being arranged in the form of 40 chromosomes. So you have a 3 million base pair genome of humans which has been arranged into 46 chromosomes, and another case you have the Paris japonica genome which has been arranged into 40 chromosomes. So you can see the level of complexity which we expect to fold such a very large genome. And all this long genome has to be arranged into a small cell to each and every cell of a eukaryote. So let's see how this is possible. If you consider the eukaryotic genome, of course it has been found into the cell nucleus. Within the cell nucleus, it mainly occurs as chromosomes at some particular stages of the cell cycle. You can see them as chromosomes. And these chromosomes, they are made up of, again, chromatin fibers. And these chromatin fibers, when they extended, they form certain regions called the nucleosomes. And these nucleosomes are again broken down. You get some proteins called the histones as well as the nucleic acids. And then finally, the nucleic acids are unbound and you get the base pairs. And since humans are being found to be diploid in nature, we have shown here 2 chromosomes. So depending upon how many a pair of chromosomes are there, like that, the depending upon that, in every organism, if it is a triploid organism, if it has 3 copies of genes, we consider it as a triploid one, and like that it will vary. The genome organization and complexity will vary depending upon how many pairs of the chromosomes are there and how much base pairs are there. The complexity will vary, but we form a basic plan for the eukaryotic genome organization, and that's what we are exploring right now. Okay, come to the level of base level of DNA. If you go to consider here, okay, this strand of DNA, every strand of DNA is being found associated with certain proteins called the histones. Histones, they are a family of small positively charged proteins which are mainly called as H1, H2A, H2B, H3 and H4. And these proteins are usually being found to be positively charged and they are being linked towards a DNA which is being found to be negatively charged. Okay, so because of this positive and negative intercharge charge difference, what happens? The binding of the DNA to this histones becomes very tight. Okay, and among these histones, the histones H2A, H2B, H3 and H4 form an octomer. What is an octomer? An octomer is something which is made up of 8 proteins. In this case, what happens? H2A, H2B, H3 and H4 in pairs, okay? 2 H2As, 2 H2Bs, 2 H3 and 2 H4s join together to form a octomer. And around this octomer, what happens? The DNA gets wound together, okay? And that, when such a thing, such a DNA combination, when you stretch it, you can find that it will be considered to be as different beads. And in between, when the stretch of DNA from these beads, okay, these beads are referred to as a nucleosomes, okay, and in between them there is a linker region, okay, and associated with that is mainly certain proteins called the histones H1. And they mainly aim in the folding of DNA into a more complex chromatin structure. And so what happens, this nucleosome bead structure here, what happens, the DNA is coiling around the surface about in an ellipsoid fashion around the histones. Okay, approximately 1 to 3 by 4 turns or 166 base pairs are being wound on the histone proteins, and before it proceeds into the next one. And this combination of histone plus DNA is called the nucleosome. And further on, you can see that when folding of these nucleosome molecules reaches a particular maximum level, the chromatin takes the shape of a visible chromosome seen in the eukaryotic cells during mitosis and meiosis. Now, the nucleosomes then are further packaged to form 30 nanometer fibers, and this is the electron microscopic view of that 30 nanometer fiber which has been obtained. So from here, when we start, you have DNA. The DNA is bound with some histones. I told you first they bind to an octomer, and then they have the histone binding is here. Then after that, they will form a 10 nanometer in diameter structure called the nucleosomes. Okay, and these nucleosomes will further again wind, resulting in the formation of a 30 nanometer chromatin fiber. And when the chromosome, mitotic chromosome is being formed, it will get a diameter of approximately 6 nanometers. So this is another depiction which I've got from the Nature Education, and it's found very understanding from base level till the end. It is quite nicely arranged. You can go through it. See, now you have a 2 nanometer DNA. Okay, if you consider the DNA double helix, it is having a diameter of about 2 nanometer. And at the simplest level, this is the chromatin is a double helical strand of DNA, and that is being complexed with histones to form nucleosomes. Look at this. Here they are being found to histones. The nucleosome is a core of 8 histone molecules, and then each nucleosome consists of 8 histone proteins around which the DNA wraps about 1.65 base times. Okay, and then you have the H1 histone here. This is having about 11 nanometer here. And then a chromatosome consists of the nucleosome plus the H1 histone. When the H1 is also being come, you call it as a chromatosome. Then further after that, the nucleosomes fold up to produce a 30 nanometer fiber. Okay, and from that, then again it gets condensed together, it gets a 300 nanometer fiber. Then this 300 nanometer fiber is again compressed and folded to produce a 250 nanometer wide fiber. That's, see, you just think this is the 250 nanometer wide fiber. And it has here, this is a diameter of about 700 nanometer also. And the tight coiling of the 250 nanometer fiber produces the chromatid of the chromosome. And finally, you get the chromosome which you have here. I hope this portion is clear to you, and let's go to the next portion in the next class. Thank you.